Hello, I'm Professor Sims, and in this video, I will discuss meiosis and sexual reproduction. This is the first in the series of 10 lessons held as part of my general genetics course. If you are a student currently enrolled in this course, please consult the syllabus and course Moodle site for assignments, quizzes, due dates, and other course information. The learning objectives for this unit include describing the behavior of chromosomes during meiosis, describing cellular events that take place during meiosis, explaining the differences between meiosis and mitosis, and the mechanisms within mitotic processes that produce genetic variation, identifying variation among offspring as a potential evolutionary advantage, and describing the 3 different life cycle types among sexually reproducing multicellular organisms. Reproduction is a fundamental aspect of life. Some examples that the book gives are hippopotamuses that give birth to calves, the Joshua trees that produce seeds that grow into seedlings, and then they talk about flamingos that lay eggs that hatch into chicks. And they give these examples because it's many kind of different ways that sexual reproduction is happening in different types of organisms. While some organisms can reproduce asexually, asexual reproduction is where they create genetically identical clones, right? Well, many organisms, both single-celled and multicellular, rely on sexual reproduction. And sexual reproduction involves the production of 2 distinct haploid cells by the parents, which then fuse to form a genetically unique diploid cell, a new organism. These haploid cells are commonly referred to as male and female, and they're produced through meiosis, which is similar to mitosis, but we'll talk very explicitly about the differences. Sexual reproduction, with its combination of meiosis and fertilization, introduces variation into the offspring, and this contributes to the success of sexual reproduction. The majority of eukaryotic organisms, including plants and animals, rely on meiosis and fertilization for reproduction. Sexual reproduction involves the fusion of 2 specialized cells called gametes. Each contain a set of chromosomes, and when these gametes unite, they form a fertilized egg or a zygote that has 2 sets of chromosomes. To prevent a doubling of chromosome sets within each round of fertilization, the diploid cell must be reduced in half. So it wants to cut its chromosome number in half, and then this reduction is achieved through meiosis, and then once the gametes are fertilized, it restores the diploid chromosome number. Let's have a look at this short video that helps to illustrate sexual reproduction and life cycles.
While sexual reproduction increases the success of a species due to more genetic diversity, it does have some drawbacks. An asexual organism can make offspring by themselves. I mean, if you talk to about anyone, you will find out that mate selection is a daunting and energy-expensive endeavor. Well, the same goes for all sexually reproducing organisms. That same energy could actually be used to make more offspring instead. Sexual reproduction slows population growth in half, as biological males are unable to make offspring. So why is sexual reproduction the method used by most eukaryotic forms? Genetic diversity! Asexual organisms can't get genetic diversity unless a mutation is introduced, whereas sexually reproducing individuals are guaranteed that each generation will be different from the last. Meiosis is the process by which sex cells, called gametes, are formed. This process maintains genetic diversity by recombining genes each generation. Mutation is still the source of all new genes, but advantageous mutations are more likely to proliferate within a species to become an adaptation due to the recombination of genes each generation. That's right, those large brains, opposable thumbs, and amazing beards are all the result of some mutation that happened many generations ago. Now, they are genetic features found in most humans, well, except beards, which are mostly due to sexual selection factors, but I digress, and we'll discuss that in the evolution chapters. In sexually reproducing organisms, which include most eukaryotic organisms, there is a cycle of fertilization and meiosis called a life cycle. Here I will discuss the differences between animal, fungi, and plant life cycle. In the vast majority of animals, they have a diploid-dominant life cycle. This means that the organism and its cells are mostly diploid. The only exception are the gametes, which are haploid. And once fertilization is complete, a zygote forms, and this starts the diploid life cycle of animals and remains so throughout life. Immediately, the cell divides and undergoes mitosis, and eventually a sexually mature adult is formed and the cycle starts over again. Now things are going to get a little bit strange, because unlike animals, fungi and algae are haploid-dominant, and that the organism is mostly made of haploid cells, not diploid, with the zygote being the only diploid part of the life cycle. Spores, which is mostly how fungi and algae reproduce, are made from the zygotes that immediately undergo meiosis. In fungi, spores develop into multicellular filaments called hyphae. Male and females don't exist in fungi. Instead, hyphae are deemed plus and minus. Incompatible hyphae are the same type, while a plus and a minus hyphae allow for reproduction. Fungi can undergo asexual reproduction by forming a filamentous stalk called a zygosporophyte, which is capped with a zygospore. The zygospore then releases haploid spores, which can make more hyphae. Now, keep in mind, this is a form of asexual reproduction. Sexual reproduction occurs by a positive and negative hyphae cross-linking together, and this forms a zygospore, which forms a diploid zygote. The only time a fungus is diploid is at the zygote stage, because immediately the zygote undergoes meiosis to become 4 spores, and the cycle begins anew. Plants are interesting as they consist of both diploid and haploid life cycles. We call this the alternation of generations. Mosses spend an equal time in both, but as you move up to more complex plants, the haploid cycle diminishes. Here I'll demonstrate using a fern. Ferns start off as a diploid zygote. This will then develop into a fern or a sporophyte, and the sporophytes then produce sporangia, which undergo meiosis to become spores. Spores grow and develop into gametophytes. Gametophytes make gametes, and some plants produce both on the same gametophyte, as is the case with ferns, and others produce separate gametophytes for each gamete. Finally, the gametes unite, creating a new zygote, and the cycle starts over again.
So we'll get into life cycles in greater detail later in the lecture, but I wanted to go back to meiosis. And here is a brief overview of meiosis. It's very similar to mitosis, but it does involve 2 rounds of cell division instead of just 1 like you'd see in mitosis. So again, I'll go through all of these steps in more detail, but for now, again, I'll go through all of the individual steps in more detail, but for now, here's a brief overview. So you have cell division that's occurring in sexually reproducing organisms. It involves 2 rounds of division known as meiosis 1 and meiosis 2. The purpose of meiosis is to reduce the chromosome number by half, resulting in the formation of haploid cells. It occurs in specialized cells called germ cells or gamete. During meiosis, DNA replication takes place before the start of meiosis 1. Meiosis 1 involves the pairing and crossing over of homologous chromosomes, and that leads to genetic recombination and increased genetic diversity. Homologous chromosomes separate during meiosis 1, and that results in 2 haploid daughter cells with duplicated chromosomes. And then meiosis 2 involves the separation of the sister chromatids, and then the result of meiosis 1 and 2 is the production of 4 genetically distinct haploid cells known as gametes. And then these gametes fuse with other gametes during fertilization, and that restores the diploid chromosome number in the offspring. So that's what it looks like overall. Right? The starting nucleus is diploid. It goes through 1 round of chromosome replication followed by 2 rounds of nuclear division. And that's how the haploid daughter cell nuclei are produced. Before I break down each individual step in more detail, let's have a look at this video that will help illustrate the overall process.
Many organisms pass their genes to their offspring through sexual reproduction. This begins when 2 gametes unite to form an embryo that is genetically unique from the parent organisms. The embryo then grows into an adult who in turn passes their genetic information on to their own offspring. Gametes are formed through a process called meiosis. The cells that undergo meiosis to produce the gametes are called germline cells. In diploid organisms, germline cells have 2 copies of each chromosome. Germline cells undergo meiosis to produce haploid gametes, which only have 1 copy of each chromosome. These haploid gametes fuse to form a diploid embryo that grows into the adult. Meiosis is just one step in the life cycle of a germline cell. Similar to mitosis, the cells also pass through the interphase, G1, S, and G2 stages before they enter meiosis. The DNA inside a germline cell is duplicated before meiosis begins during the S phase. The duplicated germline chromosomes are called sister chromatids. These chromatids remain attached to each other until the second cell division event in meiosis. There are 2 cell division events during meiosis. The first division, meiosis 1, results in 2 unique daughter cells that have half the amount of DNA as the parent germline cell. The second division, meiosis 2, results in 4 unique haploid cells that only have 1 copy of each chromosome. These haploid cells are the gametes that could go on to produce an offspring through sexual reproduction. Let's look more closely at each of the division events. Meiosis begins with prophase 1. In this stage, the DNA condenses to form chromosomes. Here we see the duplicated sister chromatids joined together at the centromere. They stay fused at the centromere throughout meiosis 1. Next, each pair of homologous chromosomes undergoes synapsis to form a complex involving 2 pairs of sister chromatids. Chromosomal material is exchanged between the 2 pairs of sister chromatids. This event is called recombination, or more commonly, crossing over. After crossing over, the sister chromatids for each chromosome are no longer identical to one another. This is one of the reasons why no 2 siblings, aside from twins, are genetically identical. There are several more key steps in prophase 1. The nuclear membrane begins to break down. Then, the 2 centrosomes migrate to opposite ends of the cell and microtubules appear. The microtubules then attach to the chromosomes. The next phase of meiosis 1 is called metaphase 1. Here, the synapsed chromosomes align at the equator of the cell. The chromosomes align randomly, which results in different combinations each time meiosis occurs. The next phase is anaphase 1. During this phase, homologous chromosomes separate and migrate to the 2 poles of the cell. Importantly, the sister chromatids remain attached at their centromeres. The final steps of meiosis 1 are telophase 1 and cytokinesis. Here, the cell divides into 2 daughter cells. Each of these 2 cells now undergo meiosis 2. Meiosis 2 is similar to mitosis. The first stage of meiosis 2 is prophase 2. Again, chromosomes condense, the nuclear envelope breaks down, and the spindle apparatus forms. The major difference between prophase 2 and prophase 1 is the fact that the daughter cells have only 1 copy of each homologous chromosome. So in prophase 2, there is no synapsis of homologous chromosomes or crossing over. In metaphase 2, the chromosomes align at the equator of the cell. Again, the alignment is random. Since the sister chromatids are no longer identical, there will be many different possible ways for these chromosomes to align. In anaphase 2, the sister chromatids are pulled apart as the microtubules shorten. Also, the ends of the cell are pushed farther apart as microtubules elongate. In telophase 2, the nuclear membrane reforms and the cytoplasm is divided into the 2 haploid daughter cells. This division is called cytokinesis. Since meiosis 2 began with 2 cells, and each of those cells were split into 2 cells, we now have 4 unique haploid cells at the end of meiosis. These cells are gametes. 2 gametes, one from a father and one from a mother, may fuse to produce a diploid embryo. The resulting embryo then grows through many cycles of mitosis.
Great, so let's dive into the steps in a little bit more detail. So meiosis 1, it's the first round of cell division, and it includes prophase 1, prometaphase 1, metaphase 1, anaphase 1, and telophase 1. And you also could include cytokinesis in that if you'd like. The first step, prophase 1, involves pairing of homologous chromosomes, creating a synapsis, forming a synaptonemal complex. Crossing over or recombination occurs during prophase 1, and non-sister chromatids exchange pieces of their chromosomal DNA during crossing over. So prophase 1 is the step where tetrads are formed. Tetrads are groups of attached chromosomes that have 4 sister chromatids. Figure 11.3 shows how in prophase 1 the homologous chromosomes come together and form a synapsis. So this guy here, the red, those are sister chromatids, is a chromosome, and then blue down here is another chromosome, and there's this synapse between them, okay? And the synapse is formed by a protein lattice made up of cohesion proteins. Figure 11.4 shows how genetic material is exchanged during recombination, or also known as crossing over, in prophase 1. So we have homologous chromosomes that are lined, and then notice that these are not sister chromatins. All of the red, these are sisters to each other. The blue, those are sisters to each other. So these are homologous, they're genetically identical. But when they are lined with chromosomes that are not genetically identical, what happens a lot during prophase 1 is that those chromosomes overlap to the point where they actually are exchanging parts of their DNA, and then they are recombined into new chromatids. So these are recombinant chromatids, this one, and then these ones here are non-recombinant. So now you have non-homologous chromosomes, non-identical chromatids. The next step, prometaphase 1, this is where the spindle fibers attach to the kinetochore proteins at the centromeres. Kinetochore proteins bind chromosomes to microtubules of the mitotic spindle. Microtubules grow from the Meiosis, that's microtubule organizing centers, and they move toward the cell's center. Microtubules attach to the kinetochores of fused homologous chromosomes during prometaphase 1, and this attachment allows microtubules to separate the homologous pair in the next phase. So by the end of prometaphase 1, each tetrad is connected to microtubules from both poles. One chromosome faces each pole in the tetrad configuration, and the nuclear membrane breaks down entirely during this phase. The next step is metaphase 1. This is a stage in meiosis where homologous chromosomes align at the metaphase plate. Each pair of homologous chromosomes randomly orients at the equator, and then the random orientation of the chromosomes determines gene inheritance in the gametes, right? So you have random alignment, you have crossing over, and then you have homologous chromosomes originally inherited as separate sets that get rearranged. And in metaphase 1, pairs of the homologous chromosomes independently line up at the metaphase plate, and this introduces randomness and variation in the gametes. It creates a diverse selection of gametes through crossover events during prophase 1 and then random assortment during metaphase 1. I'll show you this in some more detail in just a moment, but I did want to briefly discuss the last two steps of meiosis 1 are anaphase 1 and telophase 1. In anaphase 1, microtubules pull the homologous chromosomes apart, and then in telophase 1 and cytokinesis, this is going to complete the division of the cytoplasm. Figure 11.5 illustrates the random assortment that occurs during meiosis 1. So you see here, we've got AA chromosome, BB, BB, right? Then because of the way that these get separated during meiosis, during metaphase 1, you can have 4 gametes. You may end up with capital A, capital B, you might have lowercase a, lowercase b, right? But you can also end up with capital A, lowercase b, capital A, lowercase c, and then lowercase a, capital B. And this all just has to do with how the chromosomes line up during metaphase. Figure 11.6 gives an overview of all of meiosis 1. So here is prophase 1, where the spindle fibers are attaching to the kinetic core proteins, and you have each tetrad connected to microtubules from both poles. And this is also where the crossing over and recombination happens. Then during metaphase 1, this is where all the chromosomes, all the chromatids are lined up at the middle of the cell, the metaphase plate. And then anaphase 1 is pulling these chromosomes apart from each other. And then telophase and cytokinesis is dividing up the cytoplasm. And now you have two non-identical diploid cells at the end of meiosis 1. Now let's look at the second round of cell division. At the beginning of meiosis 2, the cells resemble haploid cells in the G2 phase that would be preparing for mitosis. Meiosis 2 is separating the sister chromatids within the two daughter cells. So these guys here, we want to separate those. It results in the formation of 4 new haploid gametes. In some species, there's a short interphase called interkinesis that happens before meiosis 2. Interkinesis does not involve chromosome duplication, so it's not like interphase in that regard. The two cells from meiosis 1 simultaneously undergo meiosis 2. And during meiosis 2, sister chromatids within the daughter cells separate, right? The process is similar to mitosis, but it only has one set of homologous chromosomes instead of 2. If the chromosomes were decondensed in telophase 1, they'll condense again in prophase 2. And then the nuclear envelope fragments into vesicles. The duplicated MTOCs from interkinesis will move towards the opposite poles and form new spindles. Similar to prophase 1, we have spindle formation in prophase 2. In prometaphase, the nuclear envelope completely breaks down, the spindle is fully formed, and each sister chromatid develops its own kinetochore, which attaches to microtubules from opposite poles. And then in metaphase 2, the sister chromatids, which are still held together by a centromere, are moving to the center of the cell and aligning along the metaphase plate. The microtubules of the spindle apparatus attach to the kinetochores, which is, again, attached to the spindle apparatus. Microtubules are extending from opposite poles of the cell, and the attachment of the microtubules allows for proper orientation of the chromosomes. And this ensures that when the chromatids separate during anaphase, they'll be distributed correctly into the daughter cells. Once the chromosomes are properly aligned and the spindle checkpoint is satisfied, the cell proceeds to anaphase 2, where the sister chromatids will separate and move to opposite poles of the cell. This is followed by telophase 2 and cytokinesis, which results in the formation of 4 haploid daughter cells, each containing a single set of chromosomes. So here's figure 11.6 again. This is showing the overview of meiosis 2. So it's really important to understand the differences between meiosis 1 and 2, but it's also very important to understand the differences and similarities between meiosis and mitosis. Here's a clip that helps to summarize the two, and then after you watch this, I'll go through all of the steps and compare them in greater detail.
When I was first learning about mitosis and meiosis, I learned them both separately first. And then I tried to figure out what was the same about them, what was different, why did they both matter. I would try to compare the stages by flipping through images. You know what would have really helped me? A side-by-side comparison. And that's what this video is. We assume you already have a background of mitosis and meiosis. If not, take a look at our videos on them. But this video is a side-by-side comparison, presented in a split screen. Mitosis on the left, meiosis on the right. Both of these processes, along with the cytokinesis that follows them to split the cytoplasm, are involved in making new cells. Mitosis results in body cells. Meiosis results in sperm and egg cells, otherwise known as the fancy term gametes. Before we start mitosis and meiosis, let's look at what you start with. Your starting cell in both mitosis and meiosis is diploid, written here as 2N. That means it has 2 sets of chromosomes. In humans, that's including 1 set of 23 chromosomes from mom and 1 set of 23 chromosomes from dad. 46 chromosomes total. During interphase, the cell duplicates the chromosomes. When you duplicate 46 chromosomes, you still say there are 46 chromosomes, as the newly duplicated portion is still attached at the centromere region. But there are actually 92 chromatids. Interphase isn't part of mitosis or meiosis, but it's a really important phase because it duplicates chromosomes before we get started. Just to point out, it's really hard to draw 46 chromosomes. We're going to use 6 chromosomes in our diagrams when we illustrate what's happening, because it's much easier to draw and visualize. Oh, and just a fun fact, some insects have 6 chromosomes, like mosquitoes. Unfortunately, I'm not a fan of mosquitoes, but mosquitoes do both mitosis and meiosis. When learning the stages, we give the acronym PMAT, which is helpful for understanding the stages. Both mitosis and meiosis go through these stages, but meiosis goes through them twice, and therefore has a number next to each PMAT stage. We're going to show some basic events for each PMAT stage, but please know that there is way more detail to explore than what we can include in this quick video. Prophase and mitosis. Remember that pro can mean before, and this stage comes before the others. The chromosomes are visible. We say they're condensing, which means they are thickening. Prophase 1 in meiosis. Happening here too, but the chromosomes are actually going to match up with their homologous pairs. The word homologous means that the chromosomes are approximately the same size and they contain the same types of genes in the same locations. With each pair, one came from mom and one came from dad. In this formation, chromosomes can transfer their genetic information and exchange it between each other. It's called crossing over. It can make for what we call recombinant chromosomes. Metaphase in mitosis. The nuclear envelope which had surrounded the nucleus was already disassembled before metaphase even started. For metaphase, I like to remember the M for middle, because in this stage, the chromosomes line up in the middle of the cell in a single file line. Metaphase 1 in meiosis. The chromosomes are in the middle as well, but they're still going to be in pairs in the middle of the cell, so it's not a single file line. Anaphase in mitosis. I like to think as the A for away. The chromatids are pulled away by the work of the spindles. They are moving to opposite sides of the cell. Anaphase 1 in meiosis. Same thing, but in this case, it's the chromosomes, not chromatids, being pulled away to opposite sides of the cell. Telophase in mitosis and telophase 1 in meiosis. The chromosomes are actually the complete opposite ends, and new nuclei are forming on each side to make 2 new cells. And they are starting to surround the chromosomes on both sides, as this will eventually form 2 cells. Cytokinesis follows to split the cytoplasm to complete the actual dividing of the cell. So at the end of mitosis and cytokinesis, we end with 2 identical diploid cells. In humans, they would both have 46 chromosomes. This is great for organism growth. Growing requires making more cells, after all, or replacing damaged cells. On to meiosis 2. Prophase 2. Chromosomes are condensing in both cells. It's not going to be as eventful as it was in prophase 1 because they're not going to have homologous pairs and crossing over. Metaphase 2. M for middle, but this time the chromosomes are in a single file line, similar to how metaphase looked in mitosis. Anaphase 2. Think A for away. This time, though, it's actually the chromatids that are getting pulled away. Telophase 2. Chromosomes are at the complete opposite ends, and new nuclei are forming on each side to make these new cells. Cytokinesis will follow meiosis 2 to completely split the cytoplasm. We are now finished with meiosis, and we end with 4 non-identical cells. Gametes. Males make sperm cells in meiosis, and females make egg cells in meiosis. These gametes are haploid, meaning they have the number of chromosomes as the original starting cell. In the case of humans, the resulting cells would each have 23 chromosomes. By the way, when a sperm and egg cell combine, it results into a diploid cell, a fertilized egg, otherwise known as a zygote, which will then start a series of divisions using mitosis to give rise to a brand new organism. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
So like we saw in the video, meiosis and mitosis are both processes involved in cell division, but they differ in their purpose, in their stages, and in their outcomes. Firstly, meiosis occurs in specialized cells called germ cells, and the main goal is to produce gametes with half the number of chromosomes as the parent cell. In contrast, mitosis occurs in somatic cells and is responsible for growth, repair, and asexual reproduction. So mitosis is making 2 genetically identical daughter cells. Here you can really see the difference. Where meiosis has 2 rounds of division, mitosis only has 1. Meiosis 1 involves making homologous chromosomes, pairing up, crossing over, separating, and leading to genetic diversity. Meiosis 2 separates the sister chromatids, making 4 haploid daughter cells. In mitosis, the sister chromatids are separating during anaphase, and it makes 2 diploid daughter cells. Let's go on to talk more about sexual reproduction. Sexual reproduction emerged early in the evolution of eukaryotic cells, and it's prevalent among most eukaryotes. The advantages of sexual reproduction include offspring with traits similar to successful parents, but with the ability to adapt to changing environments. Asexual reproduction allows for independent offspring production without the need for a partner, so that's why some organisms retain this ability. The asexual populations can experience faster population growth compared to sexual reproduction. Exclusively asexually reproducing multicellular organisms are pretty rare, and the reason for the prevalence of meiosis and sexual reproduction strategies are still unknown in biology. We do know that it produces more genetic diversity, and this is crucial for population survival. Sexual reproduction is continually reshuffling genetic variation, genetic information through the combination of unique genomes, crossover events during prophase 1, and random assortment during metaphase 1. In asexual organisms, the only source of genetic variation is going to be mutation. Sexual life cycles involve the alternation of fertilization and meiosis. The specific events between these two processes depend on the organism's reproductive strategy. In animals, including in humans, they have a diploid-dominant life cycle where the diploid cells, called germ cells, produce haploid gametes through meiosis. And then fertilization of the gametes restores the diploid condition. On the other hand, fungi and algae have haploid-dominant life cycles where the ecologically important part of their life cycle is haploid. Sexual reproduction involves the fusion of the haploid cells from 2 individuals to form a diploid zygote, and then that zygote undergoes meiosis to make spores that are haploid. Plants and some algae exhibit an alternation of generations with both haploid and diploid stages. Haploid multicellular plants called gametophytes produce gametes, and then fertilization results in a diploid zygote. The zygote undergoes mitosis to form a diploid multicellular plant called a sporophyte, and then the sporophyte eventually produces haploid spores through meiosis. While the specifics of sexual reproduction vary among different species, the widespread adoption of sexual reproduction provides strong evidence for the benefits of producing offspring this way. Figure 11.9 is showing that in animals, sexually reproducing adults form haploid gametes and then the diploid germ cells. The fusion of the 2 gametes gives rise to the fertilized zygote. The zygote undergoes multiple rounds of mitosis to produce multicellular offspring. The germ cells are generated early in the development of the zygote. Figure 11.10 illustrates how fungi such as black bread mold have a haploid multicellular stage that produces specialized haploid cells by mitosis, and then those fuse to form the diploid zygote. And then the zygote undergoes meiosis to produce haploid spores. And each spore gives rise to a multicellular haploid organism via mitosis. Different mating hyphae types, denoted as plus or minus, join to form the zygospore through nuclear fusion. Finally, figure 11.11 shows the unique life cycle that can occur in plants. Plants alternate between multicellular haploid and multicellular diploid. In some plants, such as ferns, both the haploid and diploid plant stages are free-living. The diploid plant is called a sporophyte because it produces haploid spores via meiosis. And the spores develop into haploid plants that are called gametophytes. And the gametes of 2 individuals fuse to form the diploid zygote that becomes the sporophyte. So that is going to conclude the Lesson 1 material. I want to thank you guys for watching. Don't forget to do the reading. Check the description for more videos related to these topics. And leave your questions for me in the comments below.